in the 15 minutes, I guess I was looking for what can I tell you about 30 years in vaccine development. What are the lessons I've learned that I might share? And essentially it's this, that if you work backwards from what makes a successful vaccine, it isn't an antigen, it isn't an adjuvant, it isn't how you deliver it. It's when you combine all of those elements together in the right fashion. And so, although as scientists, we all have our passions, and I started off life as an adjuvant developer, so obviously that's somewhat of a passion. What I learned is that's not enough, that it's the combination of all of these elements. And so if you want to be a successful vaccine developer, then you better start embracing all of these different aspects. And increasingly, as we've heard, and I think as a unifying theme, I'm a great believer artificial intelligence is, as we've already heard, going to be transformative in this process of both building the right building blocks, but then also working out how to bring the building blocks together to make a great vaccine. So we, over 30 years, essentially went from being very focused on adjuvants to then focusing on building our own adjuvant expression platforms, and I'm very much a protein advocate, but it doesn't have to be restricted to that, and trying to understand antigens. Then we realised delivery was also really important, so we then moved and started exploring all manner of delivery devices, from jet injectors to transdermal applications to oral vaccines to inhaled vaccines. And it was really, and then in the last 10 years, we've been using a lot of artificial intelligence. So it was only really when we brought all of those elements together in the one setting that we started to see real success and advancement of our vaccines. And so what I guess I'd like to show, and again, you can't do this in 15 minutes, but we believe we were able to develop transformative vaccines in a range of areas, one being therapeutic vaccines. We have a cancer vaccine that's now finishing phase 1, about to go into phase 2. We have a therapeutic vaccine for hepatitis B that's already in the clinic. We have a universal influenza vaccine platform that's looking very promising in preclinical and moving forward. We have an approved COVID vaccine, and from there we're extending that into a global respiratory disease vaccine that will protect against COVID, influenza, RSV, and human metapneumovirus that we hope to have in the clinic next year. We have an Alzheimer's disease vaccine that's just finished phase 1 trials in Australia with some very exciting data that's just about to come out. So essentially what I'm saying is, suddenly when you get the formula right, and the formula is antigen, adjuvant, delivery, and good animal models, and then a good regulatory team, suddenly everything starts to become surprisingly easy. But it was very, very difficult up to that point that we realised you had to embrace all of these challenging areas, not just one of them, to develop a successful vaccine. So in the area of adjuvants, this is still a big passion of mine. We've developed any number, I think it's probably in the thousands now, of different compounds with adjuvant activity. Obviously, most of those are still very much preclinical. We still have a screening program for new adjuvants. So anything promising that comes along, we'll develop and test. But our major focus has been on basically our polysaccharide adjuvant, where we started, which is delta-inulin, which still has some great properties. We also have a human TLR9 agonist, which is called CpG 55.2, which is in an approved vaccine and is available for anyone who's interested in a CpG adjuvant that can be used across a whole lot of different vaccine platforms. And then what we've been doing in recent years with support from NIH is to actually start combining all of these individual ingredient adjuvants to make combination adjuvants, because what we recognise is sometimes a single compound or stimulus isn't going to give us the type of immune response that we want for our vaccine. But we can do this and tailor this by combining different adjuvant compounds and different delivery systems together into the one platform. And again, I think that's really going to be the future of adjuvants, is very much not monodimensional adjuvants, but actually adjuvants that are combined and built into a delivery system. Because again, as I want to stress, the delivery is as important as the adjuvant. As we've just heard, if you want a mucosal adjuvant, then you may need to be using different delivery system and a different platform to a parental adjuvant and a parental vaccine. Similarly, at the level of protein expression, again, we simplistically 30 years ago thought, well, as long as you make a protein, they're all the same. What we've learned is they're not. So you can take the same protein sequence and you can express it in, say, an insect cell system, a mammalian system, E. coli system. They all have dramatically different effects. In some cases, some of the antigens from some expression platforms have beautiful protection, and the same antigen from a different expression platform will have lousy protection. And you say, well, what's different about them? Well, obviously, there's differences in glycosylation. I mean, that's obviously one big thing. But there are many other post-translational modifications that are occurring to our antigens that people don't even talk about, but which the immune system sees. And that can explain why the same antigen, different delivery system, and now we've got mRNA, which is a different delivery system. We've got viral vectors like the chimpaddox expression system. What you have to realise is all of those take the same sequence, they'll give you a different antigen. And again, I think it's been dumbed down and people don't appreciate those differences. And they particularly at the post-translational. You also get different cleavage of the antigens depending where they're going. And this may change their antigenicity. So you have to really think carefully about your expression platform. And then, of course, how you constitute the antigen. So it's not just about the soluble antigen or protein. It's what's the complex that you're immunising with. You know, typically viral proteins or bacteria, they form higher order aggregates. I mean, in many cases, they'll form viral-like particles, or in the case of, say, influenza hemagglutinin, if you express it in insect cells, it forms a rosette structure. And it's that rosette structure that's critical to its immunogenicity. So it's not just about the soluble protein, but what structure is that protein forming? And depending on how you express it and where you express it in different platforms, you'll actually end up with different structures. And how many people are actually studying their protein structures and trying to, again, extrapolate how that structure affects their vaccine efficacy. Now, the reason we do this is not for low-hanging fruit. Because, you know, if you take an alum adjuvant and you take a tetanus toxoid and inactivate it, you have a very successful tetanus vaccine. So again, when we're trying to get to the basic science underpinning what's happening in these systems, we're doing it for really the tough challenges. I was just talking to George at the break, and he was saying he's only interested in the toughest challenges now. You know, we've all done the easy ones, but let's look at the tough ones. And so that's where trying to think about the whole platform and the whole approach becomes absolutely critical when you're dealing with a tough problem, whether it's HIV, tuberculosis, you know, malaria. These are tough problems. And to get a solution, it's not going to just be simplistic. It's not just going to be about a better antigen or just about a better adjuvant. It's going to be about getting everything right. And so I guess that's, as I say, what I want to emphasise. Why we're doing all this science in all these different areas is to get it right so that we can tackle and succeed in the tough challenges. Now, when we talk about adjuvants, as I mentioned, there's thousands of described compounds. We've actually ourselves identified thousands of compounds with adjuvant activity. But what we have to remember, there's just a handful that are in approved human vaccines. So it's a real challenge. It's not a challenge in identifying new adjuvants. What is the challenge is how to get some of those adjuvants approved in human vaccines. It isn't easy, believe me. It took me 30 years to get my first approved vaccine with my adjuvants in it, in an approved vaccine. It took about 10 years to get them into human trials. This is not an easy exercise. So the first place to look, if you're looking for an adjuvant, is ideally go for one that's already in a human and see if it works. If it doesn't, that's when you need to start looking at vaccine adjuvants that are not yet at that stage, that are in preclinical. So yes, there still is a need for adjuvant research, but as I say, if you have something that's already there, that's much easier to use. And increasingly, as I say, what we're doing is taking our approved adjuvants and other approved adjuvants and combining them. For instance, we've combined our CpG 55.2, which is in our approved COVID vaccine. We can actually combine it with aluminium, so you get an alum CpG adjuvant, which is phenomenal if you're looking for very strong antibody responses. If you're looking for cellular responses, then you'll tend to use the CpG with other platforms, not with alum. And again, there's a whole lot of issues. Just a couple of slides to finish, just to talk a little bit about my own adjuvant. So this is delta-inulin, which is a polysaccharide particle I started working on over 35 years ago. The beauty of this adjuvant is that it's non-inflammatory. It's actually anti-inflammatory, to be precise. It suppresses inflammatory gene expression in human cells. But despite that, it's a wonderful adjuvant, as demonstrated in a number of examples I've given here of extreme examples. One is that if you take a seasonal flu vaccine and you formulate it with these polysaccharide particles, now you give it to a newborn mouse, and we're talking a 0.5 gram mouse pup at 1 day of age. Give them a single dose inactivated protein plus sugar and look at protection. And what we can show is not only are they completely protected against a lethal influenza challenge given 4 weeks later, but we actually took them to the ends of their lives with no boosters, and they were still protected, as we've shown in this publication recently in Immunology and Cell Biology. Another example of where it can be transformative is the US military were looking for how to accelerate vaccine protection. In other words, if you're naive, how do we get people protected very quickly? And what we were able to show is if we took an influenza vaccine, formulated it with this polysaccharide, we could give the vaccine after challenging the animals with a lethal dose of influenza, and they were still protected. So in other words, the adaptive immune response now outraced the virus. And so they got sick, as you can see here, just like the control mice. So it didn't stop the infection, but it actually stopped them dying from the infection, because around day 6, day 7, when they should die, they turned the corner and suddenly all recovered. And this didn't happen if you use the antigen alone or the adjuvant alone, so it was only when you use both, and we're able to show if you depleted B cells, then they lost protection. So it was actually adaptive immune protection. I thought I should have one slide on AI, because CpG 55.2, actually out of an NIH program, was completely developed by AI. It wasn't developed by me. And now it's the first human product, drug product, actually to be in an approved human vaccine. So it's gone all the way from being discovered by AI to actually being in a human product that's now being given to 8 million people. So we used, in this case, a machine learning approach. We trained it with existing TLR9 agonists and then got it to predict its own TLR9 agonist sequences, and we then screened those, and that's how we found it. So AI can be really transformative, believe me. I'm not going to talk about COVID today. Suffice to say that if you have the right adjuvant and the right antigen, which in COVID was the spike protein, turned out to be the right antigen, you formulate it with the right adjuvant, then you get enhanced B and T cell responses, and needless to say, you get protection. We've worked in many different platforms and diseases, and we're very happy to collaborate with anyone. We can share antigens, we can share adjuvants, we can share knowledge, we can share delivery systems. So anyone out there, as I say, we work through collaboration, particularly if you're working in any of these different areas. And to date, as I say, we've had our adjuvants given to 8 million people. And we have lots of human trials in progress. This is just a final slide to show how transformative the right platform can be. This is our Alzheimer's disease vaccine. As I say, it's just completed phase 1 human trials. What we're able to show in the animal models is that using the adjuvant combined with a novel antigen approach, which has the A-beta epitopes embedded in a T-cell epitope synthetic protein, we're able to induce high anti-A-beta antibodies. These were protective in transgenic models, and in fact we're able to induce antibodies that recognised A-beta aggregates in human Alzheimer's brain samples. Again, not enough time, but delivery is again equally important to adjuvants and antigens. And so thank you very much. Sorry I didn't have time to go into more science, but it's all about how you bring the package together using the right building blocks. I think suddenly you will be able to tackle challenges that so far have, proved difficult, including the malaria, TB and HIV, all of which we like George, are also trying to work to solve. Thank you.
I have a question. Thank you very much. It was a great talk. So I think from what you're describing, the packages, that's the adjuvant, the antigens, and so on. And of course, that gives you then a huge amount of complexity. You've got to somehow work through all these different combinations. So do you have any sort of lessons that you can share about in terms of how to reduce the complexity, or how you test through all these different scenarios successfully in a very short time?
Yeah, well, COVID was a great example because we had to solve all those problems in like 12 months, which was insane. It's a very good question. I think it's once you appreciate the complexity, don't run from the complexity, but embrace the complexity and build resilient structures to deal with the complexity. For instance, once you realise that this antigen structure is super important, partner with a crystallographer and get access to cryo-EM and negative stain EM and build, the tools. And we did it all through collaboration. Because again, one little group can't, do structural biology at the expert level just on a whim. So I think what we learned is don't dumb it down, don't ignore the complexity, appreciate it's incredibly complicated, and then find lots of really brilliant collaborators around the world. And that's how we developed our COVID vaccine. There almost wasn't a country we weren't collaborating with, because yes, the complexity's there. It has to be dealt with, and it can't be dealt with by oversimplifying. I think that's the danger. Thank you.
Thank you, Nikolai. So the question about you start working on the tumor or cancer vaccine. Basically, you're going to aim for more CD8 responses. So my question is that in your pool of the adjuvant, is there any group that really have a strong CD8 responses adjuvant? Second question is that although the mRNA vaccine, the adjuvant is mainly the lipid nanoparticle, do you think that for the cancer vaccine based on mRNA, adding some adjuvant further will induce good CD8 responses?
So they're great questions. The first relates to cancer vaccines. So I would view it differently. First, show that your vaccine has efficacy against cancer. Don't worry about the mechanism, because the trouble is people, again, assume mechanisms. So people assume it's CD8 T cells that are critical to cancer control. That may or may not be true. And again, it may be platform dependent. And that's what we've seen in a lot of platforms. When we pull them apart, we find the assumptions are wrong. Because, say, even with an influenza vaccine, if you modify the platform, you find actually the mechanism of protection is different. It's no longer antibodies, it's now CD4s, or in some cases it's CD8s. So let's not make assumptions. Start with the outcome, and so show your cancer vaccine is working, which we obviously did in mouse and dog models. And then work backwards and spend the time to tease out the mechanism. Now, I agree with you. We still believe that we're seeing out the outcomes we are because of mainly CD8 T cells. And the irony is that's because we know with our anti-inflammatory sugar adjuvant, we get remarkable CD8 memory responses, which again, people said wasn't possible. But we've demonstrated that hundreds of times over in many different models. So it's not surprising it seems to be also working equally well in cancer. In terms of mRNA, and you know, there is positive data already in the clinic, obviously, in combination with checkpoint inhibitors. And I think the difference, we don't use checkpoint inhibitors at this stage. We plan to in combination in the future. I think the problem with the issue with the mRNA is it's quite pro-inflammatory, and it induces counter-regulation. So it upregulates PD-1, PD-L1, which then, of course, suppress the anti-tumour response. So it's not surprising mRNA needs to be combined with a checkpoint inhibitor in cancer in order to allow the effect to be fully manifest. Whereas in our case, we've actually found just using the cancer vaccine by itself highly effective. As I say, we haven't had to add a checkpoint inhibitor. So again, it says different mechanisms or different platforms, the mechanisms are different, and therefore let's never assume we know the answer. Let's look as clinicians at the outcome and then work backwards from a successful outcome. So, but great question.